In this presentation, we'll discuss how adoptive transfer of pre-activated macrophages can be used to modulate the inflammatory process ongoing in the setting of type 1 diabetes. On the second slide, one can see a cartoon of the organization of the pancreas. On the left-hand side, the cartoon shows that the pancreas is comprised of both endocrine and exocrine tissues. In the middle you see the basic building block, the islet of the endocrine tissue, which is comprised of different cell types. On the top right you see the beta cell, which is a cell of interest, which is responsible for producing insulin in response to glucose. On the bottom panel you see an immunohistochemical staining of the pancreatic islet, which will be useful for when we see the data later in the presentation. On the third slide you see a cartoon of what's considered to be a clinical dilemma first proposed by Mark Atkinson. This picture is supposed to represent the integrity of a tissue, in our case pancreatic beta cells, and to the left hand side, when you're born, then you have a full component of the tissue. With time, if a disease process is active, then the amount of that tissue will be reduced due to the pathological processes, and the disease progresses. In the case of type 1 diabetes, the clinical debut does not occur until a large amount of the tissue has already been destroyed. And this is the problem then for clinicians, to try and see how you can protect the remaining tissue and relieve the symptoms of the disease at this late stage. So in order to understand how the therapy works, let's consider what happens in the tissue. On this slide we see a cartoon of a pancreatic islet with the insulin producing beta cells in blue in the middle, surrounded by the glucagon producing alpha cells. In the next slide, slide 5, we see the pathogenic process. A combination of pro-inflammatory macrophages and cytotoxic T cells will infiltrate into the pancreatic islets and cause destruction of the beta cells, resulting in loss of ability to produce insulin. On slide 6 you see an animation of how the therapeutic process is expected to work. To start off with, you get the destruction of the pancreatic islets, and the idea is to then introduce a large number of pre-activated anti-inflammatory macrophages, which will then migrate to the target organ. The important thing is that the anti-inflammatory macrophages in blue are far numerous than the pathogenic macrophages in red. In slide 7 you see an animation which is supposed to reflect potential immunosuppressive mechanisms. When the anti-inflammatory cells come into the inflamed pancreas, they will then deactivate the activity of the pathogenic cells. You can see that the color has changed from a bright red to a light pink. At the same time, the immunosuppressive cells which have been transferred might themselves induce other regulatory cells, such as regulatory T cells, which will also have an immunosuppressive reaction. The net effect of this will be to stop the pro-inflammatory destructive pathogenic process. In slide 8 you see what we hope happens as a final stage, and that is that once the pathogenic process has been changed, then there'll actually be an induction of beta cell regeneration, a healing response that the body will do itself, but the adoptively transferred cells will actually stimulate this process even to be more efficient and faster. So there is a possibility that there'll even be increased insulin output from the treated pancreas. So that all sounds well in theory, but does it work in practice? On slide 9 you see the abstract of a publication relating to a study in mice, in the NOD mouse model of type 1 diabetes, which was published in the journal Diabetes in 2012, where we tested this concept. In slide 10 you see the experimental design for our study. Going back to this figure that you've seen before, you can see that in the NOD mouse, diabetes clinical debut is around 18 weeks of age. Before that there's a period of insulitis where there's immune infiltration into the pancreas and beta cells are being destroyed. So we chose a time point as close to clinical debut as possible at 16 weeks of age and transferred a number of pre-activated anti-inflammatory macrophages into the peritoneum of the mouse and allowed them to wander where they wanted to. In slide 11 you see some data showing where the cells ended up. The cells were marked with a fluorescent dye so that we could use a special camera whilst the mice were living to see where the signal was. So in the left hand panel A you see just after 2 hours, and then the cells more or less where you'd expect them after being injected intraperitoneally. However, after 8 days you can see that they seem to be localized in a location which was reminiscent of the pancreas. On the right hand panel you can see what happened when we dissected the organs to see where the cells had ended up, and the majority of the cells you can see in the upper panel with the yellow-orange color, or the bottom panel with a bar graph, that most of the transferred cells we could recover from the pancreas or the draining pancreatic lymph nodes, so that ended up where the inflammation was occurring. So it's nice that the cells ended up in the right place, but did they do anything? On slide 12 you see an assessment of diabetes development. This is by measuring the glucose in the blood of mice, and in this graph it's showing on the left the amount of animals which are healthy, and on the bottom axis is the time of the experiment, age of the mice in weeks. So if we start with the red and the black lines, these are showing the untreated or the animals which received an unstimulated macrophage transfer, and you can see that within 2 weeks of start of the experiment, from week 18, the mice started to develop diabetes, and at the end of the observation time 3 months later, then 25% of the mice were healthy. This is what we normally get in this model. Not all mice will become sick. However, if you look at the blue line on the top, this represents the treated animals that received just one transfer of the pre-activated anti-inflammatory cells. And here you can see that there was a marked resistance to the development of diabetes, and by the end of the observation period 85% of the mice were actually completely free of disease. So this seems to be quite an amazing result. A single therapeutic intervention can stop animals from developing diabetes. And in these last two slides, 13 and 14, you get a nice graphic representation of this. Here we have in slide 13 the pancreas, the whole pancreas of a mouse which was not treated with the anti-inflammatory cells. And what you see in the red is the remaining insulin-producing beta cells. In the green are all the infiltrating pathogenic immune cells, and you can see that there are a lot of them and there's very little red in the picture. However, if you compare the same image in a treated animal on slide 14, then you can see that while there's still a lot of green representing a lot of infiltrating pathogenic immune cells, then there is a lot more red, a lot more insulin still intact in the treated animals. This technique is a three-dimensional imaging technique called optical projection tomography, and it shows quite nicely that the treated mice are doing a lot better than the untreated, and here we're able then to protect enough of the insulin producing beta cells so that the animal never becomes diabetic. So finally, we're kind of excited by this data. We think that this is a step in the right direction, but where do we go from here? First question will actually be, do we actually induce a regeneration effect? Is this enough to actually cause healing of the organ, or will something else be necessary? Second question is, can we combine this with islet transplantation? Islet transplantation is used in humans today, but the problem is that without stopping the autoimmune process, then the transplanted islets usually suffer the same fate as the individual's own islets and become destroyed with time. Possibly combining our immunotherapy together with islet transplantation will give an extra beneficial effect for the islets and allow them to survive for longer. Lastly, it will be good to make sure that you get as many cells as possible into the pancreas and not finding the cells elsewhere in the body, and we're working towards developing more efficient targeting strategies together with collaborators. I hope you've enjoyed this presentation and are excited by our research project as much as we are.